Welcome NEET aspirants. In this video, we will attempt question from the chapter Excretory Products and Their Elimination. So let's attempt the question now. Statement 1 says concentrated urine is formed due to countercurrent mechanism in nephron. This is correct. Statement 2 says countercurrent mechanism helps to maintain osmotic gradient. Yes, in the medullary interstitium. This is also correct. So both the statements are correct. You will have to mark option 3 as the right answer. Mark it on the OMR sheet. And we are done with this question. So this question is focused on this concentrated urine, then countercurrent mechanism, and maintain osmotic gradient in the medullary interstitium. So these are the 3 important points. And in this video, I will explain you how these 3 are interconnected. So first things first, let's check the NCERT reference. Statement number 2 says this mechanism helps to maintain a concentration gradient. This is picked directly from the textbook. And statement 1 is slightly modified. Presence of such interstitial gradient helps in an easy passage of water from collecting tubule, thereby concentrating filtrate. So the gradient is because of countercurrent mechanism and it finally helps in concentrating urine. So that's how these two are related. So let's visualize these statements, how they look in the kidney and how they work. So this is one renal pyramid. Okay. This is the cortex region of kidney and this is medulla. Medulla is in turn divided into outer and inner. The functional unit of kidney is nephron. There are two types of nephron. Cortical, which has short loop of Henle, and juxtamedullary. It has long loop of Henle. So this goes down, deep down in the inner medulla. Now parallel to this, there is a blood vessel which is called Vasa recta. It is the capillary network which is formed by the efferent arteriole. Now this is an important part in the structure of Vasa recta, this hairpin loop. This slow movement is very important for easy uptake of the solute and water. Okay, and it also doesn't disturb the concentration gradient which is established by this countercurrent system. Now these arrows, if you notice, this is going up, this is going down, this is up, this is down. Direction of movement of fluid is in opposite direction in each limb. And this forms the countercurrent system, or it is also called countercurrent multiplier. So because of the fluid movement, the solute concentration, it increases from 300 and it gradually increases up to 1200 milliosmoles. And this is the reason the countercurrent system is called countercurrent multiplier system. So the ions are trapped here and it acts like a sponge and it creates a hyperosmotic region. So filtrate from cortical nephron and juxtamedullary nephron, they both enter in the collecting duct. Now if the body requires to save water, then the posterior pituitary will release vasopressin, which is also called ADH, antidiuretic hormone. The effect of antidiuretic hormone is it will open up aquaporins. Those are special water channels in the wall of the collecting duct. So just opening the channels is not enough. You need a driving force, a force that will pull water out from the collecting tubule. That force is provided by this concentration gradient. Water is naturally attracted towards regions of high osmolarity. So the hyperosmotic interstitium ensures absorption of water. So without spending ATP, water is absorbed. So now let's focus on the countercurrent system, how it works. The aim of countercurrent system is to establish a concentration gradient in the medullary interstitium. So you need to provide ions here. The source of the ions is the ascending limb of Henle. So ascending limb of Henle has ATP driven pump. Now another important point you should remember is the ascending limb is impermeable to water and the descending limb of Henle is permeable to water. Both these limbs of Vasa recta are freely permeable to water and solutes. So with this background, now we will understand how this concentration gradient is established. So in the beginning, the solute concentration everywhere will be 300 milliosmoles. Then the ascending limb of Henle uses ATP and pumps out ions from the tubule. So these ions enter in the descending limb of Vasa recta, increasing the concentration. So via diffusion, since the wall is freely permeable, it diffuses in the medullary interstitium. This concentration difference is immediately sensed by the Henle's descending limb, and since it is permeable to water, it will lose water, H2O. And by loss of water, the concentration in the descending limb will become 400. Now see what will happen. This 400 milliosmoles will slowly move and reach in the ascending limb of Vasa recta. It will take up water and then it will reach somewhere around 350. So with every passage, little bit of extra ions and water is taken up by Vasa recta. And this is called the countercurrent exchange system. So there will be net movement of ions out from the Vasa recta. Meanwhile, these 400 milliosmoles will move from the descending limb of Henle and reach the ascending limb of Henle. Okay, so now the new concentration is 400 here. Again, it will lose ions and the new concentration will be 600. So this will be taken up by Vasa recta and then it diffuses in the medullary interstitium. In response to the difference in the solute concentration, descending limb of Henle will lose water and it will acquire a new solute concentration which is 600 milliosmoles. Now blood will move from the descending limb of Vasa recta and reach over here 600 milliosmoles and this is going to take up the water again and then move out. The next step will be 600 milliosmoles in the descending limb will reach the ascending limb. So this is how it will keep on increasing. So this will go on and on till the concentration reaches 1200 milliosmoles. It will look something like this. So the role of Henle's ascending limb is to keep adding the solute from the filtrate into the interstitium. And the descending limb of Henle keeps adjusting by losing water. The role of Vasa recta is to pick up additional ions and water and move out from the system. So this is how the countercurrent multiplier works and it helps in establishing a gradient and this gradient in turn helps in pulling out water from the collecting duct and thereby helping in concentrating the urine. So this was all that was asked in this question. Hope this video was helpful to you. Thank you so much for watching my video.